special episode of the Stem Cell Podcast, ISSCR 2024 on the ground. Hey, everybody. This is Dr. Daylon James and Dr. Arun Sharma. Welcome back to the Stem Cell Podcast, where we culture knowledge in stem cell research by talking to some of the brightest minds in the field. The Stem Cell Podcast is brought to you by STEMCELL Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. Today, we're bringing you a very special episode from the ISSCR 2024 annual meeting in Hamburg, Germany, which took place from July 10th to the 13th. It was a fantastic meeting. And we had the pleasure of meeting and discussing science with researchers from all over the world. Today, we'll be bringing you some of those conversations as we hear from delegates about their thoughts on the meeting. But before we jump into things,
whether you're looking to attend a conference this year or to expand your network, make the most out of your experience by downloading STEMCELL Technologies' collection of tools to help you prepare for your next event. STEMCELL's downloadable checklists and guides include recommendations on how to get ready before attending conferences, tips for networking, best practices for your LinkedIn profile, and more. Download the conference toolkit at www.stemcell.com/conference-toolkit. All right, let's get right into it. As Daylon mentioned, he and I attended the ISSCR 2024 annual meeting in Hamburg, Germany, and we had a great time learning about all the latest research in the stem cell field. So we also had the amazing opportunity to chat with a bunch of trainees and also attendees to learn more about their research. So without further ado, let's introduce some of the attendees that we had the privilege of speaking with at the meeting.
Hi, I'm Faye Cooper. I'm a postdoc at the University of Sheffield. The project I'm working on is developing or potentially trying to develop a stem cell therapy for a disease known as Hirschsprung's disease. So this is a disease which patients are born without an enteric nervous system. So this is the nervous system that controls the gastrointestinal tract, so your intestines, your colon. And it leads to intestinal blockages, which obviously is quite dangerous for the patient. And the only way to really deal with it currently is to remove that region of the intestine that doesn't have an enteric nervous system. So we're able to make the cells that give rise to the enteric nervous system in the embryo in a dish in the lab from stem cells. So we wanted to see whether or not could we use those stem cells that give rise to the enteric nervous system to potentially help those patients. So what we've been working on is trying to put those cells back into human tissue. So this is the tissue that's been removed from a patient with Hirschsprung's disease. We keep it alive in the lab for 3 weeks and see whether or not the cells that I've grown in a dish actually innovate and make neurons. And we're able to see that usually when you measure contractility across that piece of tissue, so whether or not that piece of muscle can contract, that the patients with Hirschsprung's disease, they're not able to contract that tissue, but when we inject our cells and leave them for 3 weeks, the tissue starts contracting again. So this looks like our cells are forming neurons and potentially trying to rescue that tissue. So this kind of is a bit of a glimmer of hope that potentially you could use those in the patient and actually try and recover their nervous system to control their intestines.
Hi, my name is Bärbel Ulmer. I'm a business development manager at ISSCR, a Swiss biotech company. We specialized on 3D cell culture models, and we especially specialize on scalable and reliable models, so 384 format. We serve the scientific community by providing with the technology and assay-ready microtissues, but we also do services for drug discovery in 3D models.
Hi, I am Ajay Kumar. I am a PhD student at INSTEM Bangalore. So we have been trying to understand the metabolic aspect of blood development. We use Drosophila as a model system. So we have this hematopoietic organ called lymph gland in Drosophila. So we try to understand how TCA cycle regulate the development and differentiation of blood cells in lymph gland.
Hi, my name is Jens. I'm a postdoc at Stanford University. I work in a CRISPR lab, but before that I was a stem cell biologist working with stem cells in the brain. And I realized that we're really lacking the technologies to program cell fate with high precision. So what we do now in my lab or in our lab is that we have developed a series of CRISPR-based tools to be able to simultaneously activate and repress multiple genes in cells. And we also developed a machine learning tool to use big data sets and predict what set of genes should you simultaneously activate and repress in a given starting cell in order to convert it to a desired target cell, while simultaneously repressing genes that specify undesired cell types. So the platform we have been developing, we hope can be really useful for stem cell researchers that are having problems making their cell type of interest. Sometimes it can be difficult to make specific cell types, especially if you want high enough purity and efficiency. And so that's the niche we're trying to slot ourselves into with this technology.
Hi, my name is Ángel de Jaime Soguero. I'm a postdoctoral researcher at the University of Heidelberg. So I am a cell biologist, stem cell biologist, really interested on cellular signaling and how this signaling impacts genome integrity across development and across lineages. So we just uncovered recently that morphogens not only dictate fate, but also can control DNA replication dynamics, as well as chromosome stability of pluripotent stem cells, for instance. So I have been now last year really researchers that are hardcore working on genome stability, on intracellular mechanisms. And they always use the same cell types, which are normally immortalized cell lines, cancer cell lines, very easy to gene edit, but to prove their own mechanism, but which lack really biological context. And I think here, people from different fields, including stem cell biology and developmental biology, can really take advantage of all the knowledge that this field has gathered to give a context on where is actually genome integrity compromised during development. And this is, I think, a new momentum also with the work with Maria Elena that will come in the next years, in my opinion. Because, especially in human development, there is still a big black box on what's going on the first 6 weeks, where 2 out of 3 embryos are lost.
My name is Maximilian Stötze. I'm from the Max Planck Institute for Molecular Genetics in Berlin. I studied an epigenetic memory mechanism in mouse embryonic stem cells, and we basically discovered how TET enzymes target specific enhancer regions, keep them methylation free, thereby enabling a transcription factor network to be established. That leads then to histone modifications that basically prime certain enhancer elements for a dormant state in embryonic stem cells that helps them to survive this dormant state.
Hi, my name is Davi Lairoleti. I'm a PI at Clever Carnivore and BMM Bio. So I'm a PI in 2 biotechs, 2 startup biotechs. BMM Bio does development of stem cell culture media and differentiation kits for human pluripotent stem cells. And Clever Carnivore is a startup that is focusing on cultivated meat.
My name is Jessica Young. I'm from Germany, and I'm a PhD student from the Central Institute of Mental Health in Mannheim. I'm currently studying schizophrenia, so regarding drug target discovery in schizophrenia, and I'm working with a co-culture model based on iPSC-derived neurons and microglia. And what I'm currently doing is I'm trying to use this model and transfer it to a high-throughput screening device and trying to use a lot of libraries to just see which kind of drug or component I can find that kind of interacts with this co-culture model. And maybe, let's see, in future, we will find some drug that might help schizophrenia patients.
My name is Ankita Kumar Bhamidipati, and I'm a PhD student in Heidelberg University. So currently, I'm in my 2nd year of the PhD program, and I work on iPSC-derived neurons, specifically in the context of bipolar disorder. So we're trying to target the prefrontal cortex and trying to generate prefrontal cortical neurons to compare patients and controls to find out any differences between the two. This can be in terms of transcriptomics or morphometrics.
Hi, I'm Indranil Singh. I'm a pre-doctoral fellow at IRB Barcelona. So basically, my major PhD work is revolving around the lineage tracing. So I have developed some tools like lentivirus-based barcoding methods, and using that, we track the stem cell clones and how do they behave if not they have the mutations. But also in this field, it has been quite hard to use these technology in human settings. So we have been working on to identify some endogenous markers that we can use to track clones inside the human beings without any editing. So we have developed a tool which is called EpiClone, and it is based on methylation, single cell methylation, targeted methylations to actually track these clones. So we started with hematopoietic system, where everyone starts with, because it's just much easier. Right now we are expanding it to the other settings like intestine or endothelial cells.
Hi, I'm Josephine Wesley. I'm a principal scientist at the New York Stem Cell Foundation in New York. I'm leading the functional genomics team at the New York Stem Cell Foundation, and I'm also utilizing the automated platform that we have at NICEF to do gene editing. The majority of our work is doing gene editing. So we generate isogenic controls from patient-derived iPSCs. We are doing gene tagging in many lines in parallel. So we have come to a point where pretty high throughput, if you want to call that for gene editing high throughput, but we can do 10, 12, 24 lines in parallel and generate clonal, cell lines and QC them and distribute them to our collaborators and partners.
Hi, I'm Carla Frau, and I am a postdoctoral scientist in Berlin in Ludovic Vallier lab. So in the lab, we study liver, in the different topics of the liver, so liver development, liver disease, liver cancer, and also cell therapy. And I'm studying liver disease and how the liver parenchymal cells interact with the extracellular matrix and how then these can be important in driving fibrosis and cirrhosis, as well as liver cancer development.
Hi, I'm Luis Oliveira. I'm a PhD student at Lund University in Sweden. So in the lab that I'm doing my PhD, we work with cellular reprogramming in the context of immunology. So my project specifically, I'm trying to understand the diversity of these very specific type of immune cells, which are dendritic cells. It's like antigen presenting cells. They are basically the ones that scavenge your whole body, and they look for antigens and they basically eat them, and then they present them to T cells and like whatever downstream response that happens from there. But they are quite diverse, and therefore there are not many approaches already available to understand that diversity. So our idea is to use reprogramming and trying by recreating, you understand. So by recreating the identity with reprogramming, you try to understand how they develop, like mirroring what happens during development in vivo.
Well, thank you to all of these attendees and trainees for participating in this episode. There's a lot of interesting research areas out there that we'd love to dive a little bit more deeply into in a future episode. So if you're interested in coming back on, send us a message. We'd love to have you.
Yeah, Arun, I 100% agree with you there. We had a great time chatting with everybody. It was really one of the things that I was most looking forward to at the meeting. Such a blast. And the speaker lineup in the meeting, the sessions, so many to choose from. We were running around like crazy. But we wanted to hear which sessions the delegates found the most interesting or what they looked forward to seeing. So let's hear what they had to say on that front.
Hi, it's Faye. So the reason why I really enjoy actually coming to ISSCR is because it's so diverse, and quite often you come across stuff that you wouldn't ever really read or see in the literature that you're usually kind of like looking for something. But for example, it was this morning, someone working on worms that you can basically chop into like 5 different worms, 5 different sections, and they completely regenerate themselves. That's amazing. But then also Elaine Fuchs, which was the last session today in the plenary session, was really, really interesting, looking at how skin cells have this memory of inflammation. And it's, really cool.
Hi, it's Bärbel. I mean, today I think it was both a bit past of myself and future. So I'm a trained developmental biologist. So I really enjoyed the session about how to use stem cells for developmental questions. But I also was in a talk by the company Maxwell, and there they showed how they think that computers can interact with brain organoids, so that you can kind of calculate or use that as a kind of computer. I don't think I understood a lot, but it sounded like Borg for me, so it was very interesting.
Hi, it's Ajay. So I have attended the talk of Professor Fiona Deutsch. I have been following her for a quite long time. I mean, even in fact, I tried to apply for a PhD position in her lab. So I really appreciated and liked her work. But that time it could not happen. Maybe in postdoc it'll help me. Still following her.
Hi, it's Jens. I'm excited about the fact that this meeting is so focused on cell manufacture. And I have really seen a shift in the last several years toward this more manufacturing side of cells, producing cells of high quality, making sure that they're good to go for drug screening, disease modeling, and eventually go into patients. And I think it's really exciting to see how the field is moving more in that applied direction.
Hi, it's Angel. So one of the most amazing presentations I have seen so far, because I didn't hear from him before, is the work of Jung-Woo on the chimeras. Definitively, that's mind-blowing. But I'm also quite fan of the work, which I already followed before, of Maria Elena, on how actually DNA replication speeds change over sulfates. So this is kind of related also to my topic.
Hi, it's Maxi. So I think so far the work of Ju Wun was pretty cool, and also Josh Beil showing the mitochondrial genes being stronger expressed in these queen naked mole rats. That was pretty cool, and also unpublished work, which I also like at this meeting, that in the concurrent sessions, especially young researchers get a chance to present some unpublished data, which I think is very nice.
Hi, it's Zavi. Today, I enjoyed seeing Dave's talk because it's a guy that I've read his papers as a PhD student, then as a postdoc, and used some of the ideas in my project. And today he was presenting on maturation. That is something that I partially worked on at Northwestern. And I'm also looking for a session tomorrow morning that will be on mechanobiology and cardiac disease. So it's a topic that is similar to what I said in my PhD. So there's a specific talk that will look at some metabolic changes in the heart and mechanobiology implications for that. And that was my PhD. So I want to see how it's done on stem cell level and compared to the things that I did 5 years ago now, because my defense was 5 years in June.
Hi, it's Jessica. Of course, I'm interested in co-culture models with neurons and microglia. So everything that's related in this sense and based on iPSC-derived neurons and microglia is very interesting to me. I already saw a lot of posters that also study schizophrenia in this sense. So that's quite interesting to me.
Hi, it's Ankita. I think the most exciting thing about the conference so far is that I get to see so many different things happening all around the world, because especially while doing my PhD, I think I've really get into this particular niche and I really forget, I don't really think about so much work that's happening in very, very different contexts and fields. So that's very exciting. It's also nice specifically with the posters, I think, because we get to interact with other students, specifically PhD students. And yesterday I found out somebody who's doing kind of exactly the same thing I'm doing, which can be a bit scary, but also nice, I think, because you get to discuss and possibly collaborate. So that's nice.
Hi, it's Indra. I think really the one thing that actually captures all of my attention, and I was like, how is it even possible, was this chimera things, which was also the part of this investigator award, young investigator award. I was fascinated by work of Hiromitsu Nakauchi lab at Stanford, and I've been closely following their chimera formation with the pig hearts and so on. So really, this was something that really captures me. But also the Matthias Luthoff talks on whole this system and the way they can form cancer-like precancerous things in a disc while maintaining all the morphology. It's just so amazing.
Hi, it's Josie. I was at a talk today from Ludovic Vallier about the next steps in cell therapy regarding liver diseases. And I know that sounds a little bit crazy because I'm doing gene editing, but in my past, I worked with iPSC-derived hepatocytes. So I'm always looking into how that field evolves, and hepatocytes are really tricky. Like, I know how hard it is. So he gave an amazing talk, first of all, and then also the progress he made to, first of all, he now has a transcription factor-based differentiation protocol, which I was hoping to see earlier. But that's great. And he really, pushed the cells to a far more mature stage, which obviously that's always the question in the room when it comes to iPSC cell therapy, right? How mature are your cells? And I think he's on a good path, which makes me hopeful and gets me always excited because I had some history with iPSC-derived hepatocytes. And yesterday I went to the diversity, equity and inclusion session, which was very amazing. And I was a little bit sad that not so many people that I was hoping to see there, were attending. But the speakers pointed out the need for more diversity in our iPSC banks, which you just don't have. Like, if you look at the numbers, it's like shocking, I have to say. And, looking back to my own history, when I was a PhD, a postdoc, I actually didn't even think about it, which is also crazy. I was not worried about, I have 2 lines, I want to study this. I did not ask a question, what's the background of this line, or like how important it might be. And if you think about it, iPSCs, when the first time we made iPSCs, the full hope was, we go away from immortalized cancer lines, which don't reflect the population, right, that we want to cure at all. It's like one cancer line that's super a weird thing that we can keep in a dish. And like the idea was, now we can have everyone in a dish, but we kind of stopped at 2, 3, 4 white European lines, to be honest, right? Like, there are a good amount of labs that expand and have more diversity in their labs, but, small labs also have difficulties to work with capacity that is more than 1, 2, 3 lines. But there's definitely a need for more diversity. And it was an amazing session. I can recommend it to everyone to watch it afterwards.
Hi, it's Carla. I was very fascinated today by the lecture of Alfonso Martinez Arias, which talked about embryonic development, but especially it touched all the ethical problems related to this and reinforcing the importance of studying embryonic development and touching all the aspects of being against the study of using human embryos. So it was really inspiring.
Hi, it's Luis. I'm really excited to present my poster because, so far we've had 1 session and I saw a lot of people and I got excited about that. I really hope that I have the same amount of people gathering around the area where my poster is. But I think that is what I found more interesting, because it is overwhelming to see these many people here when we are going for like the sessions, the presentations, but then seeing that like on the area where that you're going to be presenting your poster, it's like it turns to you, right? It's like, okay, these people might be coming to my poster. Of course, not all of them will, but hopefully some of them will.
Great responses from our delegates. Arun, what was your favorite session?
Well, I'm going to be a little bit biased. Obviously, there were a number of sessions focused on my favorite topic, stem cell research in space. I think there are actually 4 sessions on that. There was, of course, our live show, which is a huge success. Over 200 people showed up for that. We were talking with Dr. Kat Jameson and Dr. Davide Marotta about all things stem cell research in space, but there was also a really nice career panel led by Dr. Marotta, as well as Dr. Pinar Messi from Axiom Space, about how you can actually get involved in this area as a junior trainee, But going outside the space area as well, there's a really neat session. You probably remember this on cell-based meat, which is something that hasn't really popped up a lot at ISSCR before. I don't even know if it's ever been there in terms of that particular topic, but it also just is a reflection of the growth of that field. So it's just a lot of really cool stuff out there, stuff outside of your comfort zone, so to speak. So those are my favorites. But if you had to pick, Dalen, what would you pick?
Yeah, I, like you, really enjoyed our space panel that we did. It was amazing. But I'm going to go a little bit outside of the future tech that you seem to be very highly focused on, rightfully, and go back in time a little bit. I really enjoyed the Anne McLaren lecture from Alfonso Martinez Arias, which I thought gave a really great overview of the history of the ethical questions that we've struggled with and now are continue to struggle with, related to like human embryo models, but also just a nice focus on Anne McLaren herself. I thought was great. If I had to go for, the mainline session, I got to give it to Matthias Lutoff. I think he was coming in hot to this meeting, and rightfully so. And you saw the visual of all his work of the last many years, but papers that culminated in papers that were published in the last few months. That was really spectacular. And then I got to throw out a little concurrent love, and my favorite there was a talk from Kyoko Miura about the naked mole rat, which is just a fascinating model organism for so many reasons, and I think needs to be elevated. But there were a lot to choose from. You can catch up on all the sessions we attended by listening to our daily episodes from the conference, if you haven't already. We've got some more conversation with delegates in just a bit, but before we get to that,
do you want to implement the new ISSCR standards for human stem cell use in research? Get a free checklist wall chart that helps you follow the recommended reporting practices and quickly reference the relevant sections of the standards. To learn more, visit stemcell.com/ISSCR-standards-wall chart. So you might remember that back in March, we actually chatted with ISSCR leadership, Dr. Amanda Clark, Dr. Agneta Kirkaby, and Dr. Malin Parmar about what they're most excited about for the upcoming, at that time, meeting in ISSCR 2024. And you can check out that episode on our website. I mean, one thing that Dalen and I, as well as the leadership, were really excited about, I remember chatting about this with the leadership back then, was getting to explore Hamburg, Germany. And we wanted to hear more about the attendees' adventures outside of the conference center. Take a listen.
Hi, it's Faye. Do you know, I've not had a chance to look around the city yet, which I'm hoping that maybe tomorrow I'll have a bit of a time at lunch to kind of look around. But it seems really nice so far. Like, I think it helps that obviously the sun was shining when we all arrived and it's been nice and warm.
Hi, it's Bärbel. So I actually was living 6 years in Hamburg and worked here at the university doing stem cell research. So of course, for me, it's like coming home and I live with a friend instead of a hotel. And of course, a lot of people I know are here because I think almost the whole German stem cell community is here. So it feels like a family gathering.
Hi, it's Ajay. So actually, I felt like home only in Germany. It felt very natural to be here, like especially in here Hamburg. It felt like because I was in Bangalore, it I didn't see much of change, like a culture shock or anything like that. So then when it comes to commuting from place to place, it's more of like a mix of Delhi and Bangalore. Like we also have in Delhi the subway system like you have in Hamburg. So it was more of like we call it Delhi Metro. So here it's a subway system. So there also we keep on hopping from one metro to another metro to go here and there. Additionally, it's like we took a metro, this subway pass, this Hamburg pass, so we could connect through the train as well as buses also. That's a new thing which we don't have there. Like you have to buy a bus pass separately and a train pass separately. That's the difference. Yeah, we went on to marketplaces here and there. We went to port also, Hamburg port, visited there, stayed there till 11 o'clock. The shock was the daylight. It was till 9:30. So that was a very different experience.
Hi, it's Jens. I haven't been in Hamburg before, but my wife is German, so I've been in Germany several times. So I'm happy to be able to eat my Brötchen in the mornings. So that's a wonderful compliment to the science going on in this building.
Hi, it's Ansel. I work in Germany actually. So I work in Heidelberg for the last 5 years. It's my first time in Hamburg though. The venue is amazing. It's huge. I've never been, I think, in such a huge place with so many scientists around. And the city, the little that I could visit around in the last 2 days, I found it really intriguing and interesting. So I think that there is a lot of street culture activities and a lot of people just taking the sun, playing around in the parks.
Hi, it's Maxi. I've been here, but just for a transition to another city. But actually, Hamburg is a very nice city hosting this event. And in general, German science is quite fun for me so far. So I studied in a few cities. I've also been abroad. I actually like Berlin for its openness for science. It has a pretty good hub in terms of molecular biology. But I'm also probably moving on to other countries afterwards. I have in mind either Switzerland or the US, but ultimately my goal is to basically take my experiences there and then come back to Germany to become a group leader.
Hi, it's Xavi. The experience has been pretty nice. So Germany is like, people have been pretty friendly, and it's cool seeing the mixture of the different cultures, especially like if you're watching the Eurocopa, like the Euros from home, you saw like how the different populations, and we're like in one of the host cities. So like I saw a little bit of that. Yesterday, we got the chance to go to the fan zone to watch part of the game, like England versus the Netherlands, and hanging out with like people from different institutes. That was pretty cool. The city is like, it's a mix of new and old city. Because it was a city that was partially destroyed in World War II. So like you still have the Gothic architecture from like the 1500s, but you have the new things. So seeing how they merge is also something pretty nice, especially like I live in Chicago, that is a new city that was rebuilt after a fire. Here is a city that was rebuilt after the war. So seeing these contrasts is always an interesting thing.
Hi, it's Jessica. I mean, it's quite nice. It's also short distance travel, so it's quite easy to squeeze that into this busy PhD life. So yeah, it's quite nice. But I also like to experience, of course, the conferences in other countries. So maybe next year, Hong Kong.
Hi, it's Ankita. So I'm originally from India. So I moved to Europe 4 years ago, and it was quite an experience. I think it's been quite an experience. And Germany around 2 years ago. Hamburg, yeah, this is my first time in Hamburg, and I really haven't had the time to see the city. So that's the plan today, I think, in the evening. And I think, yeah, getting used to Germany took a while, I should say, because considering the language and yeah, people being more accepting of people who don't speak German, I think. But I've heard very good experiences of my friends in Hamburg, so that's nice. And I also have a couple of friends here who I plan to visit.
Hi, it's Indra. It's been incredible, like very efficient, I would say. And like, I really like the atmosphere, and a bit cold because I'm from India, at the top of India, where you have mountains and stuff. And being in Spain, it's like, it's nice to have sun all around the year, but it's also good to have some rain. This reminds me of my home.
Hi, it's Josie. Oh, I love Hamburg. It's a fantastic city. I love. We went for the lunch break to the waterfront. It was very nice. The weather is very good today. I would recommend everyone to use the afternoon or evening to sneak out for a little bit and enjoy Hamburg. It's a wonderful city.
Hi, it's Carla. We arrived 1 day earlier, the starting of the conference, and as a team building activity, which Ludovica really likes doing these things once per year, we had a treasure hunt. So we explored the city, and it was a lot of fun.
Hi, it's Luis. I come from Sweden, where currently the summer doesn't feel like summer. And I arrive in Hamburg, and the summer feels a lot like summer. And I'm Portuguese, so I'm used to like a summer feeling like summer. But I mean, so far it's been super cool. Not only the weather, although we had some rain, but also like the city of Hamburg itself. It's like it has a very cool vibe. I don't know how to describe it in a better way without using like a super common adjective, but it has a cool vibe. You walk around and you feel like you want to go places, and like people, you don't feel inhibited like trying to go to some place, especially with the language as well. Like if you don't speak German, you feel like you can go to places and still like get around by speaking in English, and that is like very inviting, and it's very very freeing, I would say.
Yeah, for sure. Hamburg had a whole lot to do. It was my first time there, and it sounded like everybody had a whole lot of fun. I mean, I had a lot of fun eating doner kebabs every single day on this trip. I'm missing those coming back to the States. You can't get a doner like that, like what you would in Germany. But outside the food, which was delicious, it was also a really fun time to be in Germany because the European Soccer Championships were going on too. We saw some couple of fun matches with folks at the ISSCR and other colleagues from England and the Netherlands who got into this really heated match that was in the semifinals. And it was cool to see the interactions between the fans and just how hyped everybody was for the match. It's an experience that I'm hoping that we get to have here in the US down the road in the 2026 World Cup that's happening here. But it was a nice flavor for soccer in Europe.
Yeah, me, I like you was on a 2-a-day program with the doner kebab, missing that as well. But I mean, I'm going to be honest, I was locked in on the meeting and very busy, so I didn't get out into the streets much. But my family was there with me, wife and kids, and they checked out the little mini model paradise where they had these massive scale models of all kinds of things with tremendous detail. Highly recommended. And besides that, we hit Gaga for the German party, got a little taste of the German nightlife in Hamburg, which was, pretty spectacular, I have to say. But for me, it was really all about the conversations in the hallways and after, as night fell, and even after midnight in some cases, with some of the scientists and friends that we've gotten to know over the years, doing the work and attending these really amazing ISSCR annual meetings. And one of the exciting things this year is that the meeting was held in Europe for the first time since 2015. This is actually the first time it's been outside North America since 2018. So given the meeting's global focus and how the stem cell research field is growing around the world, we wanted to hear about the impact of having the meeting in Europe this year and where delegates would like to see ISSCR go next. Have a listen.
Hi, it's Faye. I was lucky enough to go to the one in Melbourne back in 2018, I think it was. And that was great. And I would absolutely love to go back to that part of the world again, and potentially New Zealand would be ideal.
Hi, it's Bärbel. I'm always for Europe because I try to avoid flying. So I always take the train if possible. So I would like it to have it in distance, taking the train from Zurich. But I think something like Italy would be nice. I think that would be also that everyone brings their family to Italy.
Hi, it's Ajay. So definitely we will want to pull it to India, because right now Tina is also in the board members, not board members, organizing committee for the next conference. So I would like her to bring it to India.
Hi, it's Jens. To be honest, I was super thrilled to see it was going to be in Hong Kong. That was going to be my answer. So I'm thrilled to go there next year. I think that's wonderful.
Hi, it's Angel. Japan will be a nice place. It's true this time it was very convenient for me to come to Hamburg, directly from Heidelberg, but I think it's not about the place, but rather than about the people and the interactions that you do, I think, wherever it is.
Hey, it's Maxi. I guess Tokyo would be a nice place. Because I recently went to Japan and they have awesome science. They have really good networks, really good institutes, and also a beautiful country. So I would vote for Tokyo.
Hi, it's Zavi. Maybe I'll go for a different place. Like Brazil wouldn't necessarily be that high in the list. Would be interesting to go to Cape Town. That would be an interesting city. Try to tour interesting universities there. Stellenbosch University, Cape Town University. Johannesburg has universities also. So like, I don't know the level of stem cell research that you do, but like I've seen some papers in immunology that you have things and like other regenerative medicine aspect. So would be an interesting city and like has all the nature things in addition to the city. And you can also explore like how South Africa was built as a country. So like the history, the Mandela Revolution, the changes in the country and like how it's going developing. So would be a cool place. If it's like completely out of nowhere, New Zealand would also be cool. But New Zealand, like in addition to go to the conference, you have to take a week off to explore like everything and like see all the Lord of the Rings things and like go for a bunch of hikes. Those would be 2 cool places to go.
Hi, it's Jessica. I would probably pick Japan. I think that's quite fitting and interesting for this conference.
Hi, it's Ankita. Probably a country I haven't visited before. That would be nice because it's a good way to like see a country. So Hong Kong is pretty exciting, but maybe Japan, I think.
Hi, it's Indra. This is really a tough one, but I think I would go for Japan because they have this whole field of like developmental biology and like they have really made some nice contribution there. It would be really nice to go there and like talk to the people and like see around and do stuff there.
Hi, it's Josie. I'm very biased because I have a favorite country, so I would put it to Australia, which on the other hand, I would not like that everybody takes a plane so far away just for the environment. But yeah, I'm a big fan of Australia. I think it's an amazing country.
Hi, it's Carla. I would love Australia. So I've never been to Australia, and I think that it would be super cool to have it in Sydney.
Hi, it's Luis. If I say Lisbon, Portugal, you guys are going to like me, of course. I mean, it would be a great place for all of these people to go to because I think that most of them would love it.
Yeah, for sure. Lots of really interesting locations mentioned there. And you probably heard Japan coming up again and again and again. I wouldn't mind going back to Japan, but we're really excited to see the ISSCR go indeed to Asia, to Hong Kong in 2025. And this is actually going to be the first meeting in Asia, the first annual meeting in Asia since the 2012 meeting held in Yokohama, Japan. And who knows, maybe we'll be back in Japan in the near future. Either way, can't wait for ISSCR 2025 in beautiful Hong Kong.
All right, that brings us to the end of our ISSCR 2024 special. We had a great time at the meeting and hope that you enjoyed hearing from some of the attendees. Don't forget to check out all the episodes that we released during the meeting. You can find them at stemcellpodcast.com or wherever you get your podcasts. Until the next time, thank you so much for listening.